Okay, thank you for giving me an opportunity to talk about this work. A lot of the introduction has already been done by different speakers, so especially Trey Ilika's talk is very similar. I will try to delineate and make clear also how this is different. I would like to move this part. Very good. So here we wanted to use deep learning on biological networks themselves and train this on single cell data. So the basic idea, and we call this approach knowledge prime neural networks, the basic idea is that in a very simple algorithm, we have some inputs and we produce some outputs. So the inputs could be gene expression, and we predict if a patient is healthy or has cancer, which would be the output, and then we get those weights, obviously, and from these weights we can learn which genes are important for this prediction. Now in deep learning, these nodes are stacked on top of each other, and then the intermediate hidden nodes, they don't have any real world meaning, and we still learn weights in there, but it's hard to understand in these intermediate layers what these weights and hidden neurons could mean. And that's why deep learning and neural networks are a black box producing output from input. And so this reminded me of this analogy of regulatory biology, where we stimulate a cell, for example, and then there is something that happens in the cell and that leads to changes in gene expression downstream. And what happens in between are protein signaling networks where proteins interact and ultimately reach transcription factors which change gene expression. And so the idea behind knowledge prime neural networks is that we combine the two, that we use gene expression as the input, predict if cells were, for example, stimulated, and in between we put a network that we know of. And so here, this is where the similarity comes in. We also have some prior knowledge in this network that we put in here. But here, this network is a network of actual protein interactions. So the nodes here are proteins and not sub-processes. And so in other words, we have our prediction data, we have gene expression, so we have samples or cells and genes and values for the expression of these genes in these cells. And for each cell, we have a label. For example, was the cell stimulated or not? And then if we train classic artificial neural networks, we have a black box in here where we can't really interpret the weights that we get. We can, of course, interpret the input layer, but we can't interpret what's in between. And so I then construct these genome-wide regulatory networks which mimic biological signaling. So starting from a receptor, going down to signaling proteins such as kinases to transcription factors, which then regulate the expression of genes. And so this structure I then I use as the neural networks, and then train the network to predict the Ys, the stimulus, for example, from the gene expression. And then from in here, I can extract those weights in between and interpret the network, specifically identifying important proteins in between. And so all of this is captured in this article here. So the first important part is how to interpret or how to identify these important proteins. And so here, what I used was node weights. So the neural networks have edge weights. So for each edge, it tells us how important that connection is. It can be positive or negative, but I want to interpret the proteins. So I need node weights. And so the way I calculate them is I perturb them and then I can measure how much they affect the prediction in the end. So we can do this for each node over the different layers. We can perturb them and see how much they affect the prediction. So the nodes which are important here, those will be the ones that connect the important predictive genes to the output. And those are regulatory proteins. And to show that this works, I simulated a very simple network where I have one hidden node which is connected to predictive genes, predictive features, and a lot of other hidden nodes which are connected to non-predictive genes. And we can see that when we train this network, we get a lot of large weight for this node A. And we also see that when we repeat this training, so when we start again with random initiation, which is the basic of neural networks, of course, we always get a large node for A. So with this, basically, we have the tool in hand to interpret these networks. But I found two challenges that need to be overcome in order to really get meaningful interpretations. And so one comes from this repeated random initiation, because in theory, this could produce different results every time. And this is a problem if we have redundancy. So to demonstrate this, I simulated different networks where we have two nodes, which are hidden nodes, which are connected to informative genes, informative inputs, and to the same informative inputs and genes, and some other nodes which are connected to non-informative genes. And you can see that when we get these node weights from multiple randomly initiated networks, those two nodes get high weights, but that they are negatively correlated. And so what this means is that some of these networks have put a lot of emphasis on node B and some on A. And these are inconsistent, basically, results just based on the random initiation. So what I used to solve this problem was I used node dropout. So I dropped different nodes, visualized here, at different iterations of the learning process. And so this forces the network to split information all over the network. And so you can see that when we use this dropout, both nodes have the equal weights and they're also highly correlated. So this is the first issue, and we used node dropout. We also used node dropout not only on hidden nodes, but also on input nodes for similar reasons, which I'm not going into in detail. The second point I discovered was the effect of the network structure itself. So because neural networks are quite homogeneously connected, but biological networks are not. So there are some proteins that have a lot of protein interaction partners, and other proteins have much fewer interaction partners. And also then there is this hierarchy in the network. Some will be closer to our output, to our receptor, and some will be further away from our receptor. And this affects the node weights that these nodes get independently of any input training data. So again, I simulated some networks to demonstrate this. And here I simulate a network that has two hidden layers, the first layer and the second layer. The first layer has two nodes, A and B, and then the second layer has these 10 nodes. And you can see there's a very specific connectivity here. And again, I simulate this type of realistic data where certain genes are predictive and others are not. And you can see that based on this data and the structure, there is a certain difference in node weights, so node importance for these different nodes. And we can see that node A, for example, here has a lot of weight because it's connected to a lot of predictive genes, and node A and B, which are in the top layer, get more weights. But now how much of this is based on this data and how much is based on the network structure? So to separate this out and to normalize for the structure itself, I produced control inputs where every input feature, every gene is set to have the same level of predictiveness. So predictive means that how much does this gene by itself predict the output, and they are all set to the same level. And the weights I get from training this network on this kind of control input data, these weights just mirror this bias arising from the network itself. And so this way I can normalize and remove the effect from just the network structure, which is independent of the input data, by comparing the two. Okay, so now I have solved these two issues. First, that the results are consistent over multiple random initiations, and second, the effect of network structure. And now I wanted to try this on some real data. And so the first data set I used was the T-cell receptor signaling. So this is a data set from Christoph's lab, where T cells were stimulated by a T-cell receptor. And so we have stimulated and unstimulated cells, and then their gene expression. And so here, the first thing I wanted to validate was this idea of the dropout. And we see that when we increase the dropout, the rate of dropping nodes randomly in the network, we decrease the prediction performance, which is generally very high. So in this task, prediction performance was very high, also with alternative methods. And we increase the correlation of node weights over randomly initiated networks. And so I selected this network, this dropout of 10%, where the prediction accuracy is still quite high, but also node correlation already has seen a lot of benefit here. So in summary, this network can predict very well. And also we identified a number of proteins in here through our interpretation method, so using the node weights. And these were highly enriched for known T-cell receptor signaling proteins. Interestingly, these proteins were not changed in their gene expression. So downstream, the input gene expression of these same genes that code for these proteins was not changed. So we really get those intermediate hidden proteins out, which would not be reflected in gene expression data, and therefore would also not be found by any kind of feature interpretation, because the gene expression is the features, and any interpretation on the feature level would not have revealed the same kind of interpretation. So this is, of course, very interesting, because these proteins are really the regulatory mechanisms how the gene expression is linked to the phenotype, and that's really what we want. So then we wanted to see, okay, how robust is this, and how does this is affected by this control data? And so what I performed next was a shuffling experiment where I take this network and I completely perturb the network by random shuffling, but keeping the basic properties, so how many neighbors each node has, equivalent. And that's shown in the next slide. And then I looked at this enrichment of annotated T-cell receptor regulators on the x-axis. On the x-axis, you see this regulators enrichment, and the distribution shown is the distribution over these shuffled networks. And what you can see is that if you just use the node weights based on real data without any control, the enrichment is the same, or approximately the same in the shuffled networks than in the real network. This is also true when we just use control inputs, which are again these completely artificial inputs just for control. But when we compare the two and we calculate what I use are differential node weights, so comparing real data and so the node weights based on real data and node weights based on control inputs, then I really see a strong enrichment for T-cell signaling regulators in our approach. So this really highlighted, the shuffling experiment really highlighted the importance of controlling also for the network structure. The algorithm used for network shuffling is this one, which took me quite some time to find, because it's actually not straightforward to create these hierarchical shuffled networks. Okay, in the following, so now we have also shown that this approach and these two methodological optimizations are really useful for interpretation. Now I want to show also the versatility of this approach in larger and different data sets. The first one is the human cell class. So this is a large data set of about 500,000 transcriptomes. And here in two organs, bone marrow and cord blood, I used KPNNs to predict immune cell subsets, so T cells, B cells, and monocytes from gene expression. And then interpreted the intermediate layers. Now this network is not a network where I have this one receptor, like the T-cell receptor, but here I use multiple different receptors to really simulate all kinds of signaling pathways that could occur in a cell. And then again, I get out these regulatory proteins and their importance, and found that in the bone marrow, those were very much related to developmental biology and pluripotency as expected from bone marrow, while in the cord blood, I saw more mature processes like T-cell receptor signaling, B-cell receptor signaling, and so on and so forth. I think we're going slightly over time, so I'll keep this short. But basically, I have also applied KPNNs on a time scale. So here we have a different, this is a data set from acute myeloid leukemia. And this acute myeloid leukemia, there can be these AML cells can come from different stages of hematopoietic development. So they can be more similar to hematopoietic stem cells or progenitor cells or myeloid precursors or monocytes, for example. And I trained KPNNs at each of those stages to predict between the AML cells and the healthy cells and identified regulators important at the different stages. Or in a second data set where we had glioblastoma, another cancer of the brain with different subsets. So there are different types of glioblastoma, there's 4 different types. Here again, I trained different knowledge-primed neural networks to distinguish regulator importance in these different subsets. And in both cases, at least anecdotally, I could show that these definitely recapitulate a lot of what is known about the regulation and some additional proteins that could be interesting. So in summary, this approach of knowledge-primed network enables us to somehow put cell signaling interactions into hidden networks, into neural networks, going from receptors to signaling and kinases to transcription factors to gene expression, and then extracting those hidden nodes. I have outlined some challenges and the solutions we found. And again, all of it is in this paper and more. And with this, I thank, of course, everyone in the BOC lab who has helped me with this analysis and discussed with me. The SEM and the Institute in general, the Vienna Scientific Cluster in Vienna, where I did a lot of the calculations. And again, I just started a group in September in Salzburg. Thank you. That's it from my side.
Thank you. We have time for one or perhaps two questions. The first question is by Vitaly Kleshchinov. Oh, complicated. Apologies for not pronouncing your name very properly. Kleshchevnikov. Vitaly Kleshchevnikov asks, how do KPNNs related to causal inference, in particular with regard to redundancy? Wouldn't you expect that kind of redundancy in the network is causing quite a few challenges?
We use the causality based on prior knowledge. So the network has a certain structure which is external. So people have characterized signaling interactions, and they have characterized which protein signals to which other protein, and this is the causality I use. So I don't infer causality like the first speaker of today, so not like Jonas. But I use the causality that is inherent to the data that I use to build the network. The redundancy, so that I've shown that there is redundancy, and the network, if there is redundancy, it's hard to distinguish which one is the, if two nodes, hidden nodes, have the same input features, it's hard to say which one is more important. And so there I use dropout to solve this problem. I hope I answered this question.
Great. Final question. We also saw Trey's talk earlier. Kind of, do you see ways how these two approaches could be integrated, and what could be gained from integrating these two approaches?
So the first thought is this difference of using proteins and interaction network, and in Trey's work it's more groups of genes which are in the same biological processes. One could, of course, think of first somehow starting with a network and then converging into some processes, for example. I think that the idea of gene expression and mutations, so mutation would be Trey's work and gene expression is more this work, of course, has been discussed. This is also an interesting area for further work. So yeah, I think those are the two main lines where this could be done.